Back. So we've taken a quick look at some microtubule-based motors and some microfilament-based motors, both of which can generate movement in cells. And what we're going to do is kind of look at the microtubule-based motors and see how, the microfilament-based motors, and look to see how those myosins work with actin to produce muscle contraction. We're not getting into this in too much depth because physiology covers it in quite some detail, but we'll take a look at it briefly. So this is so muscle contraction is probably the most familiar example of mechanical work that's mediated by intracellular filaments, so the cytoskeleton, and motor proteins. Now, thinking about muscle, mammals have skeletal, cardiac, and smooth muscle. Now, we're not going to look at all types of muscle, we're going to look at striated muscle. So that's primarily skeletal muscle. So skeletal muscles are responsible for voluntary movement, so all the things that we do, which are conscious movements that help us maintain locomotion. So if we think about a muscle, that consists of parallel muscle fibers. So if we took a piece of muscle out, we'd see this bundle of muscle fibers here, and those are muscle cells. And so each of these fibers, each of these cells is a long, thin, highly specialized multinucleate cell. So each of these cells have multiple nuclei. And if we look at the fiber, each fiber, so if we take one of these fibers, one of these cells here, so this is a cell, here's a nucleus, is what we see is that there are numerous myofibrils. So here is the next unit if we go to the next kind of scale down. So there's a myofibril, and then each myofibril is divided up into structural units called sarcomeres. And then the sarcomere contains bundles of thin filaments, which are blue in here, and those will be actin. And the actin the thin filaments are obviously made of actin, and there's some other proteins there that we'll talk about a little bit later, troponin and tropomyosin. And then there are the thick filaments, and those are made from aggregates or bundles of myosin proteins. So that's the basic structure of muscle. Let's take a look at the myofibril in a little bit more detail. Is what you see is that the filaments in skeletal muscle are all aligned, which gives these myofibrils this alternating pattern of dark and light bands, and we'll see this a bit more clearly in a moment. And so those striations are characteristic of cardiac muscle and skeletal muscle, and they're called striated muscle. And so the dark bands, which you'll see in a moment, are called the A bands, and the light bands are called the I bands. So when we take a look at this structure in a moment, we'll see dark bands, the A bands, and light bands, the I bands. But if you took a cross-section through the myofibril, we would see these arrangements of thick filaments, so that's myosin, and then we've got these thin filaments, which would be actin and troponin and tropomyosin. And so then when this is arranged and we look at it in 2 dimensions, cut the other way, rather than cut this way, if we cut that way through the image, then we would see the dark bands and the light bands. So let's take a look at those. So here's our dark band, that's the A band, and then there are these lighter regions flanking the A band called the I bands, those are the light bands. The lighter region down the middle of each A band is called the H zone. So this region of light here in the dark is called the H zone. Okay, so that's the H zone. In the middle of each I line, or each I band, I guess, is this darker band called the Z line. And we'll talk about the structure and the function of that in a little bit. And so the distance between one Z line to another Z line indicates a sarcomeric unit. So the sarcomere is from this Z line here to this Z line here. Okay, so that's just a lot of nomenclature so that we can talk about what's going on. So let's just review very quickly. We've got thin filaments, those are actin, troponin, tropomyosin. Thick aggregated bundles of myosin, so those are the thick filaments. Then we've got Z line, that's part of the I band, and then that that's flanked on the other side by another I band with a Z line, and in the middle there's this H zone with an M line running down the middle. And we'll talk about these all structurally in a moment. So the arrangement of the thick and the thin filaments in myofibrils gives rise to the striated appearance that you see here. The striations are dark and light, and it's also responsible for the observed shortening of sarcomeres during contractions. Because what we're going to get is, if you think these are two sets of filaments, we're going to get the myosin to slide the actin in one direction. So we're not going to walk in two directions, we're just going to slide the filaments over one another, and because they're anchored at each end, everything's going to pull in and we're going to get contraction. So let's look at the thick filaments, then we'll look at the thin filaments, and when we're done with this video we'll come back and we'll talk more about the mechanism of contraction. So right now we're just doing the structural pieces. So each thick filament, here's a thick filament, consists of hundreds of molecules of myosin, and one half of the thick filament, the heads point in one direction, and the other half of the thick filament, the heads point in the other direction. So here's that bare zone, that H zone that was in the middle of the last diagram. So if we go back and look at that diagram, here is that H zone, and so our M line would be right down the middle here, and so these myosin point in one direction and these myosin point in another direction. And just think about it, because what you're going to have is the myosins basically pulling in opposite directions because they're aligned oppositely, and that's going to pull the sarcomere together. And so you can see that the myosins are all arranged in this staggered fashion, so that the head groups protrude at different points, and they will eventually form interactions with the actin in the thin filaments, in which case they become cross-bridge head groups. So the thin filaments are going to interdigitate, so that means they're going to overlap. So we'll have a thick filament and then a thin filament and a thin filament and a thick and a thin. So they overlap. Interdigitate means overlap. So let's go back to looking at the thin filaments here. So when we look at the thin filaments, we've got three main components here. We've got F-actin, which is in blue here, which we've talked about before, filamentous actin, the diagram at the bottom and a model at the top. So F-actin, and then F-actin is kind of stabilized by triple myosin, and that's going to become important when we look at contraction and how myosin attaches to the actin. And then we have troponin here, and troponin's got three components: TNT, TNL, and TNC. And this is like a little molecular switch, and that molecular switch is responsive to calcium. And so it's this part here which causes a change in this filament that allows the myosin to actually do the contraction part, to actually move the actin. And so this is going to be sensitive to calcium, and so we'll talk more about the role of calcium when we get there, and we talk about the mechanism of contraction. So the actin in the thin filaments is oriented, so here's the thin filament, thick filament, thin. So this is interdigitation that I mentioned, so the thin and the thick overlap, but not completely, sorry. The actin in the thin filaments is oriented, so the plus ends are anchored at the Z line, so there is our Z line, here's our M line, so here's one end of the sarcomere, and then we would have another end of a sarcomere over here somewhere. So the plus end is anchored at the Z line, and that's done by a cap Z. We'll talk about that in a moment. Myosin 2, so these are the myosin proteins, these are myosin 2 molecular motors. When they move, they're going to move towards the plus end, so they are going to walk towards the plus end. But because everything's anchored, is what's going to happen is things will contract, things will slide as the myosin walks towards the plus end. That's going to pull this Z line towards the middle, towards the M line basically. So we're going to get a sarcomere this long contracting to this long, and that's going to give you the muscle contraction. There are a number of structural proteins here that contribute to holding everything in place, I guess. What have we got? Let's find alpha-actinin. So here's alpha-actinin. That keeps the actin filaments bundled together, and that's what makes up the Z line. So the Z line is primarily made of alpha-actinin. Cap Z holds the plus end of the actin filament to the Z line, so that's holding the actin to the Z line, and then the Z line kind of bundles all of the actin together and keeps it in place. Tropronodulin here on the minus end, the filament maintains stability, and then there's a protein nebulin here that wraps, and that maintains the integrity of the actin filaments. We've got myomycin. Where is myomycin? Here. Myomycin sitting there is present in the H zone, so that's that kind of clear zone that we looked at, and that's going to be bundling together the myosin. So everything's being held in place by these structural proteins. And the last one, titin. Titin is one of the biggest proteins. I'm not sure if it's the biggest, but it's one of the biggest proteins encoded in the human genome. And so titin is basically going to anchor the myosin thick bundle to the Z line, and that keeps the myosin bundles in the correct position relative to the thin filaments when you do contractions, so that everything's kind of held and locked in place. So a lot of structural proteins. Make sure you can go back, and I'm not worried about these structural proteins too much. I'm more worried about you understanding the role of the actin filaments and the thick filaments and the troponin and the tropomyosin, which we're going to come to in a little bit. Okay, so I'll be back soon with Muscles in Action Part 2.